Hi, I did my reading assignment and on open source knowledge discovery platform. The summary of the paper, the below the in summary of the paper, we had the reference of the paper. The below three are the references of the paper, and coming to the purposes. mm, Atomic Unlimited built a free programs, free programmers, SIM to facilitate the use of machine learning methods on high dimensional biological data such as that generated by the transcriptomy, flow symmetry, or the microbiome. With Simon's help, we are able to do this. Simon aid even the least technical savvy biomedical researchers in locating patterns in their data and constructing high quality prediction models by integrating a graphical user interface, pre-existing pipelines for predictive modeling and automated machine learning. This is possible because of the machine learning capabilities already presented in Simon. Coming to the related literature with the profilatory use formulation of a large and complicated disease, the life and the health sciences have placed a greater emphasis on the relevance of data analysis and the discovery of new information. Most doctors and the biologists sadly lack the technical programming abilities and knowledge of algorithms needed to do such research with the goal of streamlining the use of over 180 cutting edge machine learning algorithms and high dimensional biological data. We have created Simon, a modular open source program. Thanks to its intuitive graphical user interface, standardized pipelines, and automated algorithms for machine learning and other statistical analysis approaches. Simon is a tool that helps both non-technical and technical researchers uncover relevant patterns in biological data. Simon is a powerful resource for identifying the best algorithms, and it also helps in locating the best algorithms. A research designer strategy previously an unforgettable amount of data may now be collected at levels of detail made possible by advancing data generation techniques. High dimensional data integration may help biomedical scientists learn about previously undiscovered biological systems and develop accurate and preventive projections of a clinical consequence. Inadequate low quantity analytical tools may be to blame for executingly slow phase at which new information is exchanged among biological researchers. Built open source software we have built open source software package called Simon tool that scientists employ machine learning on a high dimensional biological data. Simon's intuitive graphical user interface and standard automated machine learning approach make it possible for even non-technical researchers to quickly and easily find patterns, extract input information, and build reliable prediction models in high dimensional data. As Simon automates and standardize these evolutions, it can do them. It can do them. Results, discussions, and conclusions. Genomic sequencing, high dimensional flow symmetry, mass symmetry, and multiplicity imaging are only a few of the recent technologies advances, but that have produced a dueling hues and integral biological data. Since this is the case, data extraction now requires complex, complicated methods. Since scientists are always on the lookout for innovative, computational approaches to handle the vastness and diversity of the data. The goal of machine learning, a subfield of artificial intelligence, is to train computers to make inference about the future based on the present and the past. To name just a few, ML has been very helpful in following areas: genealogy, proteomics, cellular image analysis, drug discovery and development, and phenotyping. This has resulted in a paradigm shift in our appreciation of the complexities inherent in biological systems. Precision medicine analysis, a patients' genetics, transcriptomic and proteomic patterns at a systems level to um, foretell how they will react to a treatment or how their condition will proceed over time. Recently, ML has become the computational approach of a choice for this kind of therapy. The most difficult pattern is figuring out how to put machine learning principles to use, and the second hardest part is actually putting those concepts to work to get insights. Coming to the contributions, even second by bioinformaticians have difficulty completing these labor incentive ML activities due to absence of an uniform methodology for the successful executions of machine learning operations. And this is due to the fact that operations include so unavoidable danger besides ML. These are the codes, and many in the life sciences research community cannot make use of these libraries because they lack graphical user interface. Also, they are useless unless the user is proficient in R or Python. This is why there has been a recent uptick in efforts to standardize these libraries and build a framework that would make ML deployment more approachable in life sciences. And coming to critical analysis, in critical analysis, we have the oral assessment. In oral assessment, we're discussing the oral assessment. Mm, Familiarity with a variety of conceptual libraries that can be used for data pre-processing and cleaning, data segmentation, model development, and tweaking assessment of the model performance and reduction of overfitting is essential for analysis of large data sets and the extraction of knowledge using machine learning. And coming to the R and the Python has become the FATO standards and statistical computer in recent years. Each language has been extensive usage in the development of powerful applications that perform the that perform the aforementioned operations. As a result of their popularity and the fact that they are free to use, R and Python have attracted enormous communities of developers who are continuously improving and extending their libraries accessible to users. They are now 15658 distinct R packages available on the CRN uh, repository of May 2020. Many of the available programs have radically different syntaxes when it comes to training a model, creating predictions, and giving weights to variables. And in research methodologies, these these and other factors are contributed to a recent upstick in activity pre-processing of partition modeling, evaluating partition modeling, evaluating um, partition modeling for biological data sets with a high percentage of high percentage for biology, high percentage missing values and uneven distributions of such as many in infected subjects and a few health controls or both. Specialized software is required to overcome the cues of dimensions or poor predictive power, as seen when the number of features is significantly greater than the number of samples. 38. The machine learning method is only one part of what the app can be accomplishes. It should also be to do exploratory analysis and provide the results in an understandable graphical user interface. Increase in both the quantity of biological data sets and the amount of processing power needs to analyze them may be tracked back to the fast growth of technology due to its importance in today's data centers running on the cloud computing. Open source web-based software has been freely available for the research so that you may conquer these obstacles. We develop a Simon. It's a Simon is actually is called as a sequential iterative modeling overnight. On the Simon program was developed and it's freely available to the public with the goal of expanding the usage of machine learning to the life sciences. It reduces the time needed to develop high quality ML models, freeing up researchers to devote more time to discover and less to unkeep. With its unified interface, Simon simplifies the creation and evaluation of prediction models using different machine learning techniques. To know 182 unique ML approaches are available to streamline the whole machine learning process. The create library automates of feature selection, model creation, and evaluation. This frees up extra expert machine learning user to concentrate on a data pre-processing, feature engineering, and relaxing their models. The newly knowledge learned, the development of study hypothesis, and the collective of data may fall within the preview of a subject matters expert. At the individual may lack the technical abilities required to efficiently exploit machine learning. Additionally, Simon offers a consolidated strategy for training models, adjusting hypermeters, and evaluating models by means of development of training, validation, and test sets. This aids the model's ability to generalize to new data and avoids to optimistic accuracy predictions. In order to objectively access the models, a test set, known as a holdout set, is employed after the training set, and a tenfold cross validation has been applied to them. We hope that many people as possible would benefit for using replicating our models. Hence, we provide downloads of them in our data format. In addition to the conventional machine learning technique, the pre-configured software has a number of characteristics that made it useful for analyzing more biological data. Clustering significance can be visualized. Genes can be tested over or under representation, and data with many missing values can be integrated using the multi-set intersection function. For good measure, Simon acts as a both client and a server. And from this paper, the questions I need to discuss is: are there more areas where we can apply this? And the second question is: what are the data set we can use here? These are the questions I need to discuss from the paper. And thank you.